Welcome everybody to a Striatech hosted journal club. My name is Thomas Münch and I'm one of the founders of Striatech. I welcome you to another of our journal clubs where scientists present their projects and data, highlighting the use and the application of our products. Striatech is a dynamic biotech company from Tübingen in Germany. We specialize in neuroscience testing tools, in particular for vision research. The Striatech founders are all experienced neuroscientists in the field of vision research. Today, it is my pleasure and my privilege to welcome Rachael Pearson as our speaker. Her talk is entitled Photoreceptor Cell Therapy to Treat Advanced Retinal Degeneration. Rachael is Professor of Developmental Neuroscience at King's College London and co-leads the Ocular Cell and Gene Therapy Group based in the Center for Cell and Gene Therapy. She is internationally recognized for pioneering work on photoreceptor transplantation and stem cell-based therapies to restore vision in inherited retinal degenerations. After studying natural sciences at the University of Oxford and completing her Ph.D. in neuroscience at the University College London, she built her career at the UCL Institute of Ophthalmology before moving to King's. Her group has published several landmark papers showing that transplanted photoreceptors can integrate into the retina and recover visual function in preclinical models, helping to lay the foundation for emerging clinical approaches. Rachael has received numerous honors, including a Royal Society University Research Fellowship, the Alcon Research Institute Award, and election as a Fellow of the Royal Society of Biology. Before you start, Rachael, let me remind the audience that you will be able to ask questions throughout the event and also to give a thumbs up to other questions. You will get to those questions at the end of Rachael's presentation. When you want to ask a question, the cursor by default is in the name field. Giving your name is optional, but you have to click into the question field before you start typing your question. The Q&A panel on the right-hand side will stay available for a couple of days after the live event. So if you're currently not watching live, but if this is a recording of the event and if Q&A is still available, please make sure to enter your question and your name so that we can back to you with the answer. We're very much looking forward to your questions. So Rachael, please go ahead with your presentations. Don't forget to unmute your microphone to share your screen with us. And I'm very much looking forward to your talk.
Thank you. I'll just share my screen now. Okay, with a bit of luck, you can see my slides now.
Absolutely, yes, thank you.
Okay, well, thank you very much, Thomas. Thank you for the lovely introduction and for the invitation to be part of this seminar series. So it's a great pleasure to be able to present some of our work over the last couple of decades, actually looking at cell-based therapies for the treatment of advanced retinal disease. So this is work that, as Thomas already summarised, that I've done primarily at UCL and King's, and this has been part of a collaborative effort with my long-standing collaborator, Professor Robin Alley. So just to start off, I thought I'd begin with a quick schematic just to orientate everybody. So I'm just going to move my little thing out of the way. So just to start off the quick schematic so that orientate everyone. And so today's talk is going to primarily focus on the light sensing neural tissue of the eye, the retina, which in this schematic is shown in yellow. So just going to pull up my pointer, apologies. So always the technical starting bits. There we go, laser pointer. And also thought it was worth pointing out the macula, which is a highly specialized area within the retina, located pretty much opposite the pupil and the lens. And whilst this region accounts for only around 1% of the total area of the retina, it contains the majority of the retina's population of cone photoreceptors. So there are two types of photoreceptors in the eye, rods and cones. Rods mediate a lot of light under dim light conditions, and cones are particularly important for daylight and colour vision, and they are tightly packed into the macula, and within it an even more specialised structure called the fovea. And this very tight density of packing brings about the ability to mediate high acuity vision. So that's the ability to see fine detail, reading, recognize faces, and so on. So just now let's turn our attention to what goes wrong during degeneration. So retinal degenerations are some of the leading causes of untreatable sight loss in industrialized countries. So retinitis pigmentosa, which is shown in the bottom right of this fairly classic example of vision loss, RP is an umbrella term encompassing inherited forms of degeneration affecting rod photoreceptors primarily, but this can lead to a secondary loss of cone photoreceptors as well. And so because of the impact on the rods, you have this loss of peripheral vision and an encroaching sort of tunnel vision that can actually eventually encompass the whole visual field, affecting the central vision as well. And then conversely, inherited disorders affecting cones obviously have greatest impact on the macula and high acuity vision. And an sort of classic example of a sort of multifactorial macular disease is that of age-related macular degeneration. This is a complex disorder where age is the biggest risk factor, but this combines with environmental factors like smoking and obesity, and there's also a hereditary component as well. But despite all these very different underlying causes, these diseases share a similar sort of common feature in common, which is the progressive degeneration of the photoreceptors. That said, the remaining retina, whilst it undoubtedly undergoes changes in response to this loss of photoreceptors, it remains broadly intact. So that inner retinal circuitry, so shown here in the schematic up at the top. So you have the second order neurons, the bipolar cells, and then those connect to the ganglion cells, which obviously is the output of the eye via the optic nerve to the higher visual processing areas in the brain. So it's this inner retinal circuitry that remains largely intact. And this, of course, then presents us with the opportunity to potentially restore vision and at least reverse that loss of visual function by transplanting healthy photoreceptors in place of the diseased ones, in expectation that they can then form new synaptic connections with the remaining retinal circuitry. Now, neuronal replacement anywhere in the nervous system is undoubtedly challenging, but the eye and perhaps also the macula within it represent an exciting and potentially feasible target. The cells that we are trying to replace are afferent neurons, they're sensory neurons, so their input is light, so we're not trying to recreate input, incoming synaptic connections, and they only need to make a single synaptic connection to the bipolar and horizontal cells in a triad synapse in order to feed into the remaining visual circuitry. And if we think about the macula itself, it's a relatively well circumscribed area of around 1.82 millimeters square in area in the human retina. So we're not trying to repopulate the entire retina, but perhaps a sort of functional patch is a feasible target. That said, whilst photoreceptor transplantation is potentially conceptually quite straightforward, there are still many aspects that we have to consider when we're trying to develop a therapy beyond the simple and indeed not so trivial, could it work? We have to think about how we're going to get our cells in. We need to think about the type, what is the optimal donor cell for transplantation? Whatever that cell is, we need to achieve connectivity between it and the remaining retinal circuitry. We have to, the degree of connectivity has to be sufficient to bring about both retinal and visual function. Can we improve function? So ultimately with the cell therapy, we want it to be as efficient as possible. We want as many of the cells that we are transplanting to form new connections. So can we improve the sort of rate of synaptic connectivity? I've included breadth of application here because cell therapy is often presented and advocated as a potentially a disease-agnostic approach. So if you consider that something like gene therapy, for example, which, as you know, has been seeing great progress in recent years, the sort of challenge with that is that obviously you have to develop a new therapy for each individual gene that's affected. So there is quite reasonably significant interest in trying to develop more disease-agnostic approaches that could have one therapy for a number of different underlying diseases. And actually, if you've been following the seminar series in December, Connie Sepko gave a beautiful seminar looking at a gene therapy-based disease-agnostic approach, where trying to introduce things that would prolong the survival of the remaining photoreceptors So again, a sort of neuroprotective strategy style. So again, looking at something that is one product that can treat many diseases. And cell therapy sort of sits within that bracket as well, because obviously we are ideally making a one product, which is the photoreceptor, and then transplanting that into potentially multiple different diseases. And then from there, we've got, obviously, we have to then think about things like GMP manufacturing, scaling up manufacturing, and clinical trials. I'm not going to focus particularly on these aspects in today's talk. I'm happy to discuss them in questions. I thought I'd focus on the other aspects first, but this is work that we are actively working on at the moment. Okay, so photoreceptor transplantation approaches generally fall into one of two main sort of ways of getting your cells in. And as I said, this really is more about the mode of delivery than any mechanistic differences in terms of how they'll rescue visual function afterwards. So we have cell suspension approach and tissue grafts. They're fairly explanatory in their names, but just to explain in a bit more detail. So cell suspension, we take donor material, that might be fetal retina, it could be retinal organoid-based material, dissociate it into a single cell suspension. And from there, you have the ability, if you want to, depending on protocols, you can then sort the cells of interest. And this is certainly the strategy that we've worked on a lot. So the advantage is that you can have this ability to select the cells of interest, so in our case cone photoreceptors, and get rid of everything else. So this is advantageous in terms of being able to reduce other cell types in the retina, like inner retinal cells that may potentially impede integration. It also means that in, for example, an organoid culture where you may not have a uniform population of retinal cells, you may have other non-retinal cell types present, you can get rid of those as well. So you can have a highly purified, highly defined population of cells that you can transplant through this method. The surgery is relatively straightforward. We can administer them through a very fine gauge needle, similar to that for gene therapy. So the sort of surgical trauma is quite limited. We can introduce them into the subretinal space, which is this region just here in pale pink. So that's a pseudo space between the remaining inner retina and the overlying retinal pigment epithelium. So we can put them directly in the point of contact. Now, a limitation of the cell suspension approach is a loss of relatively poor organization. You're effectively, as I like to call it, putting in a cell soup. And so the onus is then on the cells that you're transplanting to have sufficient information on board, intrinsic information, to be able to self-orientate, so demonstrate polarity, send out a new axonal projection to form synaptic connection, and outer segments in the opposite direction. So the tissue graft approach helps to get around this to a degree because you're putting in a more organized piece of tissue. So again, this could be taken from fetal. So some of the pioneering studies, Magdalena Zeiler and Robert Araman did a lot of this, looking at taking fetal tissue and transplanting that. More recently, they and others have worked with retinal organoid derived tissue. And again, the same principles apply. You're sort of taking a piece of intact tissue and then transplanting that again into the subretinal space. So exactly the same approach. So this has the advantage that your tissue is already reasonably laminated and organized, so you can potentially put it the right way up, if you like. Depending on the stage that the tissue is in terms of its maturation when you transplant it, they can go on and form rosettes. So although they go in laminated, it doesn't mean that that laminar structure is always maintained. So this is obviously an area of development at the moment. And another challenge is that they come with inner retinal neurons. So certainly this is the case with the fetal tissue, and even with organoids, there are still inner retinal cells present. So you can see that these could potentially cause a challenge for your donor cells. Your donor photoreceptors have to disconnect from the cells that they come in with and move around that in order to then form new connections with the recipient retinal cells. This is sort of work that's been investigated. Michiko Mandai in Japan is doing fantastic work looking at this approach, and is also looking at genetically engineering the organoids to reduce the number of inner retinal cells to try and circumvent that problem. Obviously, with tissue grafts, there is more complex surgery. You're putting in a piece of tissue rather than a fine gauge needle, so there are considerations around that. And then there's an approach for sort of bringing the two ideas together, so using bioengineering. where we might take donor cells that have been isolated, as I described earlier, and then combining this with biomaterials that might create a sort of organized structure in which to seed those cells, a little bit like the outer nuclear layer. And so there are several groups that are looking at this as well. So it's sort of a hybrid between that purified cell population and that organized structure. So all of these approaches are under active investigation at the moment, and as I said, they all have pros and cons in terms of sort of how to get that connectivity and organisation. I'm going to talk to you mostly from here on. I'm just going to focus on the cell suspension approach. That's what we have focused on the most in our group at the moment, although we're looking at the others. This is the furthest advanced, so I'm going to just talk to you about that for the remainder of this talk. Now, before focusing on recent progress, I just wanted to give a bit of a sort of historical context regarding some of the earlier studies, both by us and others in the field, and how this has shaped our understanding over the years of the different mechanisms that underlie rescue. So around, alarmingly, 20 years ago, when I was first working on this, much of the published data concerned taking different types of stem cells, adult, embryonic, mesenchymal, for example, and transplanting these into models of retinal degeneration. And apologies in advance to grossly summarize a large body of work in about 1.5 sentences. A lot of these studies found that although you could transplant the cells, they failed to correctly differentiate into retinal and particularly photoreceptor phenotypes. So we started to look at this problem when I joined working with Robin, and we started to do looking at mouse-to-mouse transplantation, and we took donor cells from the peak of photoreceptor birth, so around neurogenesis, and we took sort of immature, more mature photoreceptors at different stages and transplanted these into adult recipient retinas. And what we've found was that the best donor cell was a post-mitotic photoreceptor precursor cell. So this is a cell that has exited the cell cycle, so it's no longer proliferative. It's committed to a photoreceptor fate, but is yet to fully mature. And it seemed to be that this hit the sweet spot, because if you think about what we're trying to do in transplantation, we are transplanting an immature cell into a fully mature environment, and we're asking it to recapitulate a developmental process, which is synaptogenesis and continue its differentiation pathway in the elaboration of segments as well at the other side. And it seemed to be that taking these precursor cells, these cells were sufficiently immature to survive the undoubted sort of stress of dissociation and transplantation, but sufficiently mature that they could complete, sufficiently differentiated that they could complete the rest of that maturation post-transplantation, even in the context of the adult environment. So this, and I'll describe some of these studies in a bit more detail in a couple of slides, but this then presented the next challenge, which was how to generate donor cells for transplantation from renewable sources. So those studies were done in using mouse postnatal tissue. And obviously, if you to translate that into human situation, then that's the equivalent point in development is during the second trimester. So we would then obviously turn our attention to how to generate stage equivalent donor cells from a renewable source. And obviously that then falls to stem cells, so embryonic stem cells and induced pluripotential stem cells. So the challenge, as always, with stem cells is knowing what cues to give them in order to turn them into the cell type that you want. In our case, the photoreceptor, or perhaps the RPE cells, for example. And here I think that there are many reasons why I think working in the eye is cool, and we're very lucky. But I think that this is one example of when, as a working translation, how we have really benefited from being able to work within the eye, because it has long been the model of choice for developmental biologists because it's an accessible model of neural development. And so because of this, we know a huge amount about the signals, extrinsic and intrinsic, that are involved in making a stem cell into a neuron, and more importantly, a retinal neuron, and within that, specific subclasses like a photoreceptor. And so a lot of early investigations, both our group and many others, focused on traditional 2D culture methods using embryonic stem cells and iPS cells. and trying to provide external cues to guide them towards this retinal fate, and progressed so far, but generally failed to get fully differentiated photoreceptors that exhibited all the hallmarks that we would expect. And the big breakthrough really came through with the work of Yoshiki Sasai's team, and the advent of 3D retinal organoids. So this is when you're taking ES cells or IPS cells, but differentiating them within a three-dimensional context rather than in the traditional 2D. And from here, we start to see this sort of spontaneous generation of these 3D retinal structures. And this really sort of transformed, I think, the field in many ways, but certainly for transplantation, it suddenly meant that we had a way of generating sort of bona fide photoreceptors. So our protocols are variations. So there are multiple published protocols by many different groups available that use variations of 2D and 3D stages of differentiation. Our own protocols have been developed for various purposes, primarily as a sort of factory for the manufacturing of cone photoreceptors for transplantation. And then we have different protocols that better reflect the normal retina, and particularly the lamination that we tend to use for disease modeling and the study of retinal development. Okay, so we can now generate both mouse and human photoreceptors from stem cells. So let's return back to the transplantation itself. And again, I'm just going to go back to some of the earlier studies, just because there are some important aspects to note again about this mechanisms of rescue. And this is dependent very much on the stage of degeneration that you're treating. So in our initial proof of principle studies, we were seeking to transplant either into adult wild-type animals or models of partial degeneration. And the rationale at the time was that we thought this would be easier. You'll see why in the next slide, why this turned out to be a bit of a curveball for us. But we thought that this would be a more straightforward challenge for transplantation than going straight to advanced disease. And the idea was that if we transplanted into a model where there was still some outer nuclear layer left in that host retina, then there was cytoarchitecture within which your donor photoreceptors could migrate into and integrate within and then form new synaptic connections. And so we thought that this would provide the transplanted cells with a better structure and greater chance of survival before we then move the goalpost to more advanced disease. So the donor cells, we started off looking at rod transplantation simply because we were working in the mouse at the time, and we had transgenic models of labeling photoreceptors, particularly post-mitotic rods in this case with GFP. And alternatively, you could use viral vectors. But regardless, when we transplanted, we saw large numbers of these very beautiful GFP-labeled cells within the host outer nuclear layer. And importantly, these cells also expressed the proteins that were missing in the host photoreceptors. So remember, these are models of retinal degeneration. So in the top example that you've got here, this is the rod alpha transducin knockout. So it's a GNAT1 knockout, which encodes for rod alpha transducin. And you can see that the GFP positive cells all express rod alpha transducin labeled in red. And then here in this middle, in the bottom panel in the middle, you have the RDS model, so retinal degeneration slow. This has a mutation in peripherin 2, and you can see that the green cells are expressing peripherin. And the same goes for on the right-hand side, rhodopsin knockout. The green cells are expressing rhodopsin. So the donor cells, the healthy donor cells we're transplanting into these disease models are all expressing the proteins that are missing in those host retinal cells. So this was all highly indicative that the donor cells were migrating into the recipient structure. And indeed, when they're present in sufficiently large numbers, we get rescue or visual function. So we could do single cell recordings of the GFP labeled cells and show that they had light activated photocurrents. We could do optomotor. I'm very sorry, Thomas, this was in an earlier version of the optomotor. This wasn't a Striatech machine, and this was a very labor intensive version of it. But we did see improvements. What you'll see subsequent to this is the Striatech machine. But this earlier iteration, we did still see improvements in optokinetic head tracking in the transplanted animals. And we could also see improvements in water maze activity and even restoration of activity in V1 of the visual cortex. And this compared with sham injected animals and untreated, where we saw no such rescue. So this was all looking very promising. This all seemed great. Except that when we then started to switch our attention to cone transplantation, which was obviously most relevant for human vision, we started to see some anomalies in what we were seeing after transplantation. And so we were starting to transplant donor cone photoreceptors, this time again GFP labeled, and we'd transplant them into the retina. Only this time we kept on seeing examples of cells that looked very much like rod photoreceptors, not cones. We considered the possibility that perhaps there was some transdifferentiation. We were putting these cells in at a sort of relatively immature stage. Perhaps there's plasticity and they could switch fate because they were in a rod-rich environment. So we then transplanted a population of cells that were genetically engineered so that they could not turn into rods. So we knocked out NRL, the transcript and the downstream transcriptional pathway for rod differentiation. And we still saw cells that looked like rod photoreceptors. So at this point, there was a somewhat confusing and mildly stressful period of research where we started to try and understand what on earth was going on. And the upshot of that research was that we identified a very unusual mechanism that was going on, which we termed material transfer. And we published these findings back to back with Marius Adder's group, and then this was very shortly followed afterwards by Robert McLaren's group, Valerie Wallace's, Ivana Zenovich's as well. So this was a sudden sort of shift in our understanding in the field about how the mechanism of rescue was occurring. And what was happening is that the donor cells are able to exchange material with the remaining host photoreceptors, material transfer. And this is a process that allows the exchange of proteins, both cytoplasmic and membrane bound, and it's remarkably efficient, sufficient to rescue function in those accepting cells. Now, this occurs in mid-stage degeneration, and this is because there are still photoreceptors in that host retina available to receive material from your donor cells. We then went on and investigated the mechanisms the cellular mechanisms that underlie this rescue, and similarly ended up publishing in parallel with Valerie Wallace's group. And we both found that actually what's happening is that the donor cells are forming these membranous sort of nanotube-like connections, so these sort of cytoplasmic bridges between the donor and the acceptor cell. And this allows a shuttling of material from the donor to the acceptor. And this is enough to both its GFP, but it's also proteins such as rod alpha transducin, rhodopsin, peripherin, and so forth. So all these things are going across and rescuing those sick cells. Now, this is only in situations where there are still photoreceptors alive to accept that material. So in parallel, I should also point out that there is true integration that occurs alongside. So our original hypothesis in the field had been that the donor cells were migrating into that remaining outer nuclear layer and integrating. You do see this, but it probably accounts for around about 5 to 10% of the actual total number of cells that you see, GFP label cells that you see. The majority are there because of material transfer. However, in parallel, we had then moved on to looking at advanced end-stage degeneration, because this is obviously more relevant from the clinical perspective, because we are likely to be going in at very advanced stages of disease for cell therapy in patients. So we'd already turned our attention to advanced disease, and in this case, material transfer is no longer relevant because there are no photoreceptors in the host retina to receive anything by material transfer. So the only way that rescue can occur in advanced disease is by donor-host synaptic connectivity, so the donor photoreceptors connecting to the remaining inner retinal circuitry, the bipolar cells and the horizontal cells. Okay, so we now have example of transplantation into the RD1 model of advanced disease. So this is a classic retinal degeneration model. Photoreceptor loss is extremely rapid. Most of the rods and cones are lost by about 1 month of age. And staining at the bottom left, as you can see, confirms that there's a complete absence of cones in the central retina in this example. We were actually transplanting in this model at 3 months of age, so much further on, and then looking 3 months later, so 6 months of age. From here on, everything that you'll see is human stem cell, embryonic stem cell derived cones transplanted into mouse models. So no longer looking at mouse, this is all human cells into mouse models. So this just shows you, this example here just shows you an example of a small region of a transplant, we can readily repopulate areas of around 1.5 to 2.5 millimeters squared, so similar to the size of the macula. And the transplanted cells, you can see, are repopulating an area of the outer nuclear layer. And that if you look at the cells labelled in white, these are the bipolar cells of the host that are beginning to send up processes towards the donor cell transplant. And if we look at the ultrastructural level, we can also see evidence of formation of presynaptic terminals. You can see here in this panel with the ribbons, so electron dense bars of the ribbon synapse. And actually, if you look carefully, you can see sort of nice synaptic vesicles lined up along them. And on the left, we can also see starting sort of nascent outer segment formation. So these are relatively early on, and I would say that actually subsequent transplants, I think as we're defining our donor cell population better and better, we're getting sort of better maturation and transplant outcome. But nonetheless, even with these are sufficient to drive improvements in retinal and visual function. So first up, we just have a simple light-dark avoidance test. So mice are nocturnal, they're prey animals, so they'll preferentially seek out dark spaces over well-lit ones. The box has 2 chambers and there's a little door, and they can freely move between the 2 chambers. And on the far left, this one, panel E, you can see activity for a untreated RD1 mouse. So you can see that there's no preference between the 2 chambers, and that contrasts with the amount of activity sort of spent tracking it in panel H. This is a wild type mouse. You can see he spends much more time in the dark box than in the light box. And then in the middle 2, you have 2 examples of transplanted RD1 mice. So on the left in panel F, this is one that's received a transplant of human cones, but they were derived from a patient with achromatopsia. So these are iPS cells rather than ES, but they are from a achromatopsia patient, so they are completely non-functional. So in every other way in their differentiation, they're identical to the normal healthy cells, but they're because of non-functional, they provide us with a really nice control for neuroprotective potential neuroprotective effects, or and also for material transfer as well. And as you can see in this model, or this example rather, there is no difference between the time spent in either of the 2 chambers. And conversely, in panel G, this is a RD1 mouse that's received normal functioning healthy cones, and they spend much more time in the dark box in preference to the light. We can then look at the retinas in more detail, so sacrifice the animals and look at the retinas explanted, and we can perform electrophysiology on them using a system called multi-electrode array analysis. So this is a series of electrodes, and we place the retina down on the electrodes such that the ganglion cells, so the output of the retina, are in contact with those electrodes. And then we can record the activity, the spiking activity in those ganglion cells in response to light shone at the retina. And on the right-hand side, you can see 2 examples of this. The top one is a retina that's received functional cones. The bottom is one that's received these cones from the achromatopsia patient. So on the bottom, you can see there's no light evoked activity. You just have spontaneous firing from the ganglion cells, which is indicative of a normal, happy, healthy retina. On the top one, you can see that instead with these guys, that on presentation of a light flash, we get a significant uptick in the spiking activity of the ganglion cells in the region of transplant. So the whole retina is exposed to the light stimulus, but we only see activity in the region of the transplant itself. We can then further investigate the nature of the responses by spike sorting. So this is a process where we look at the individual sort of waveforms of activity of the ganglion cells picked up on each electrode. So each electrode can measure more than one retinal ganglion cell. And then we can look at the individual ganglion cell profiles and see what they like responding to. And encouragingly, we can see a mix of different response types, including on, off, and on-off, indicating that the parallel processing pathways are still functional after transplantation. We do see a preponderance of on at the expense of on-off responses. There are a couple of caveats to this. You know, our stimulus that we're using in these experiments is a light step from dark. So it's darkness and then a bright flash and then off. This will probably bias towards detecting on type responses. And we're now trying to move towards more naturalistic stimuli and looking at different background intensities to start to really unpick this in more detail. It may also reflect the anatomical observations of what is going on during degeneration. So Rachael Wong and colleagues, for example, did a very nice investigation in the primate retina, doing a laser ablation, and showed that the on pathway seems to be maintained for longer than the off pathway, for example. So there seems to be different stability in the different types of pathways in response to injury. So this needs further investigation. But either way, we still see on and off processing, which is really encouraging. So then we decided to move the goalpost a little bit further. So this is now transplantation into the aged RD1 mouse, this time transplanting at 12 to 15 months of age and then looking 3 months later. So this is work in revision at the moment, which we're hoping to publish soon. And so this is still the RD1 mouse, but this time by going much later, this was to try and address this idea of what is the sort of therapeutic window for transplants. So I've said the likelihood is we were going to be going in quite late. If we're thinking about things like age-related macular degeneration and frankly also long-term RP as well, then we have to know that we may be needing to treat quite late on after a lot of these changes and this sort of plasticity and remodeling has occurred. So whilst I said at the beginning that the inner retinal circuitry remains broadly intact, that sort of tagline is true, but it's not entirely true, or it's not the complete story. So although the bipolar cells and ganglion cells and so on don't degenerate to the same extent as the photoreceptors, there is a bit of attrition and there is remodeling. So we know, for example, the dendritic arborization of the bipolar and horizontal cells completely retracts. There is substantial remodeling as to how those cells try to connect. And we also know that the molaglia will undergo reactive gliosis in response to that degeneration. They can become hypertrophic, they send up and they start to sort of ramify their processes up along the apical surface, which might then create a physical barrier between where we're transplanting our cells in the subretinal space and the inner retina to which they need to connect. So transplanting much later, so this is now over a year after those photoreceptors died, was to really see does that host retina still retain that plasticity to respond to healthy photoreceptors? Really encouragingly, we see that, yes, it does. So if you look at the middle row in particular, this shows secretagoguin labeling, which will label cone in the mouse retina. It labels cone on and off bipolar cells, and PKC alpha will label the on bipolar cells. So that's rod and cone on bipolars. And you can see that in both cases, we get re-elaboration of the dendritic processes up towards the donor cells. And we also see the same thing for calbindin positive horizontal cells as well. So despite it being over a year since they had photoreceptor input, these cells respond to the presence of new photoreceptors coming in. And also with the GFAP labeling, this labels the reactive monoglial population. And actually, we see quite extensive remodeling of the molaglia to the extent that they appear to actually extend processes up above the donor cell mass, almost in an attempt seemingly to incorporate it within the new retinal structure. So this was very encouraging. We also can do synaptic staining. I would say that synaptic staining in the donor cell, cell suspension approach is quite challenging because the synapses don't necessarily form a nice, neat antiplexiform layer like they would in the intact retina. These synaptic-like contacts seem to appear a little bit spread around the transplant. But nonetheless, even at this advanced stage of degeneration, we still get improvements in retinal and visual function. So this is back to the MEA recordings again. Along the top, you can see an untreated aged RD1 mouse. There's no light evoked spiking activity. In the middle row is a GNAT1 mouse. So this is the rod alpha transducer knockout that actually I mentioned earlier on in the talk. In this case, it's now being used as a cone-only positive control. Because the rods are non-functional, the cones, but the cones that do function, it gives us quite a nice sort of comparison for cone-only function. So the middle row shows you GNAT1, cone-only mediated function, and at the bottom you can see the human cones transplanted into the old RD1 mouse, and we get improvements in retinal function again. And we also see improvements in optokinetic head tracking. This is the Striatech system. And actually, I think it's possibly sort of worth sort of commenting at this point just about the use of optomotor in these sorts of transplants, because unlike in gene therapy, for example, where you might be able to transduce pretty much at least half, if not the whole retina, cell transplantation, you're transplanting a small focal patch. And so then in a test that involves freely moving animals that have no instructions to pay attention to what's on the wall, I think we find that sometimes that we can see good responses on some trials and not on other trials. And I think that's more to do that we just have a small patch. And unlike in a patient where you could ask them to attend to a certain direction, sometimes that's obviously not possible with the mice. And so we get very good responses on some trials, and occasionally then the next time it will apparently not see. But on the untreated, we never see any responses. Compared to then in black, this is the GNAT, this is the cone only, so effectively cone mediated sort of normal function. So then moving on to a different model. So this was begin to try and address this idea of it being a disease agnostic approach. So this time, this is the AIPL1 model of Leber's congenital amaurosis. So for those of you not familiar with this is an incredibly rapid model of degeneration. The photoreceptors start to die before they've even begun to form that first synapse between themselves, bipolar cells and horizontal cells. So these retinas do not receive sort of light-mediated function. And so this seemed to us like quite an interesting sort of very severe case to see if we could try to rescue with photoreceptor transplantation. And we actually see, despite the fact that these cells or these retinas hadn't formed a normal sort of first triad synapse, first vision synapse, they do seem to be able to respond to the presence of these donor cells. So we have a nicely organized donor cell mass showing some degree of polarization, and we see elaboration of the dendritic processes of the bipolar cells and also the arborization from the horizontal cells as well, so along here at the bottom. So the host retina is responding to the presence of these donor cells. And again, much like we saw with the old with the RD1 transplants, we get restoration of light-evoked retinal activity. So the same format applies here. So we have the top is an untreated AIPL1 mouse. Again, no light-evoked activity. Middle shows you the cone-only wild-type sort of control response. And at the bottom, you've got the human cones transplanted into the AIPL1. And again, we get nice micro-ERG traces and good, robust activity correlated to the light onset and offset. And once again, we get improvements in optokinetic head tracking. We can also look at different aspects of the vision that we're restoring or visual function that we're restoring. So we can play around with different light levels. So what I've shown you so far is from a darker adaptive background and a full light flash. We can also do light flashes of increasing intensity. And we can see that if you compare on the left-hand side, the GNAT1, so the cone only, versus the transplanted, you can see that the responses kick in at a very similar light intensity between cone only, intact retina and the transplanted, and that the amplitude of responses increases in line with increasing intensity as well. And then on the right-hand side, we're also beginning to then play around with contrast sensitivity. So instead of going from black to bright, we can also have light adaptive backgrounds, and we can also do different steps. And we can see that different ganglion cells are showing responses to different contrast sensitivities. The top one just gives you an example of a particularly sensitive ganglion cell showing high contrast sensitivity. It responds to quite subtle differences between background and light stimulus. The one at the bottom has lower contrast sensitivity, and it is quite a marked difference between background and light flash. So another aspect that we were keen to investigate was around sort of spatial mapping, and this goes back to this idea about the organisation or potential lack thereof after transplantation. And actually this applies both to the cell suspension approach and the retinal sort of sheet graft approach. Because if you think about in the normal retina, a lot of the ability to map a visual space is brought about by the retinotopic map, and that's that a photoreceptor's position in the retina dictates its contribution to our visual space. And particularly with areas like in high acuity vision around the macula, for example, you have a very tight association between a photoreceptor and a bipolar cell immediately beneath it and a ganglion cell immediately beneath that, and this can almost be sort of 1-to-1 ratio. Now, in transplantation, we no longer have control about how that arrangement is formed, whether or not that photoreceptor really connects to a bipolar cell immediately beneath it, because the donor cells could send out axons in different directions, and even the host bipolar cells could, when they're re-elaborating their dendritic processes, we have no control about whether or not they are also going to maintain this sort of apical basal polarization. And what this could mean is that you could have light being detected by a photoreceptor in one location, and it's actually sending it down to a ganglion cell some distance away from it. So to begin to probe this, we started to present the transplants with spatially structured stimuli. In this case, this is a series of just light bars, horizontal and vertical. And then we can start to correlate the retinal ganglion cell activity relative to the position of the bar. And you can see that on the left, bottom left of the histograms, this is just a sort of cumulative histogram showing that the ganglion cells located on the activity being detected by the electrodes immediately beneath the light bar, you see the highest activity, and this falls away very quickly compared to electrodes and ganglion cells at increasing distances away from that stimulus, from the position of that light bar. And actually in the transplants, we see something very similar. Again, the highest spiking activity is immediately beneath the light bar, and this falls away quite quickly on either side, compared to the untreated, where obviously there is no spatially defined responses. So it seems to indicate that we're at least preserving a degree of spatial mapping. We're now probing this further with more complex stimuli, looking at receptive field mapping and so forth. So this is work that we hope to be able to publish in the near future. And this sort of brings me on to just the last bit and last couple of slides to talk about improving connectivity and organisation, because, as I said at the beginning, ultimately for transplants, we want as many of the donor cells that we're transplanting to connect up to our host retina, and to do so in as organised a manner as possible. And we sort of stumbled across an observation. One of my PhD students, Anna Malati, she was looking at connectivity and trying to do tracing experiments to try and sort of understand this connectome in more detail, but actually sort of stumbled across another interesting observation. Everything I've shown you so far with the human transplants is 3 months post-transplantation. This was slightly lighted on by chance, and it worked, and one of those typical things that if it ain't broke, then don't fix it, and you carry on with that same process. But we also looked at a few time points earlier than that, and in this case this just shows you a 6-week post-transplant. This is actually for completely different experiments, but then serendipitously what Anna observed was that actually the horizontal cells labelled in calbindin had already started to elaborate new arborizations out from the inner retina towards the donor cell mass at 6 weeks. The bipolar cells, however, had done absolutely nothing and were still quiescent at this point. And you contrast that with the 3-month time point, where you now see quite a lot of extensions from both the bipolar cells and the horizontal cells. And what's striking about this is if you look at this compared to normal development, this is sort of mimicking a normal developmental sequence. So in development, the first synapse forms between the between photoreceptors, and not with bipolar cells, but with horizontal cells. So photoreceptors connect to horizontal cells, and then the bipolar cells come in secondarily to then form this triad. And so what is quite intriguing is whether or not we are actually recapitulating a normal developmental process, even though this is in a fully adult environment. Obviously, our donor cells are primed to be at the stage of forming axons and synapses. perhaps the host retina is also then going through this similar sequence of similar developmental sequence. So this also started to get us thinking about whether or not we could then try and manipulate the host environment to make it even more developmental-like. In particular, try and put something in that could introduce sort of apical basal polarity and perhaps a sort of an attractive cue for our donor cells to tell them which way to go to form the synapses and send out their connections. And so we did an in vitro screen of a variety of different candidates that we thought were particularly interesting, looking at transcriptomic publications. I'm afraid I'm going to be a little bit coy and just call it candidate A, because this is unpublished data, and I'm quite excited about this at the moment, and we're still doing quite a lot more work and investigation. But just to show you that we've identified a potentially interesting candidate that seems to act as a chemoattractant for human cones. It generates more neurites and longer neurites. And if we reintroduce this molecule into the host environment, it also seems to promote that same polarization in our donor population after transplantation. And then looking at the functional output, although this is quite preliminary at the moment, the transplants that have been transplanted into retinas that have also been primed with our candidate molecule seem to show a much stronger spiking activity, more robust, sort of tighter firing compared to our control transplants. Whether this is better polarization, whether it's bigger, so more synapses, we don't know at the moment, and that's work ongoing. But at the moment, it's very encouraging to see that we can actually improve transplant over and above the baseline. So in summary, human stem cell derived cones can rescue both retinal and visual function in multiple models and stages of advanced retinal disease. So we hope and really think that this now is sort of providing good evidence that it represents a disease-agnostic treatment for retinal disease. The transplanted retinas exhibit a range of responses across physiological light levels and exhibit spatial information processing. Again, more work is needed to really sort of understand this and sort of probe this further, and that's ongoing at the moment. And the sort of first hints that we might be able to actually improve on our standard sort of baseline of transplantation and start to think about whether or not we can mimic processes involved in normal development in order to do so. And with that, I will just I have undoubtedly missed people off because there are an awful lot of people involved in this work, but just enormous thanks to everyone. It really does take an army to grow the cells and do all of these transplantation work. It's a huge effort, and particularly thanks to the Medical Research Council, who have underpinned our work for many, many years, and obviously several of the sight loss charities, Fight Macular Society, Retina UK, and we've also been very fortunate to have great support from the Wellcome Trust Ph.D. schemes over the years as well. And with that, I will stop, and thank you for your attention.
Thank you so much, Rachel. That was a tour de force for real. So we have quite a few questions already, so let's dive right in if you don't mind. So we'll start with a more general question. What do you think are the main impediments for implementing cell transplantation as the go-to therapy for retinal degeneration or inherited retinal degeneration? And realistically, how long do you think until we get there to have this as a therapy?
I love the how long question. It's always those. I think that so at the moment, I think that the evidence coming through from us, from other groups, I think that there is an increasing body of evidence to say that it works, that we can restore or certainly improve function. I think that now, obviously, ultimately, we need to see efficacy in clinical trials. But I think probably in terms of the sort of main impediment, it's probably around scale up, I think. So, and this is something we're actively working on at the moment, because obviously you're manufacturing a living product and you've got to then do that at scale. What we can do, where we were starting off sort of even 5 years ago, the number of cells we could generate versus what we can generate now under GMP conditions, and to be able to then look at all of the regulatory testing that needs to go on before you can put that into patients. That requires a significant scale up, which we've achieved. But then there's the next level of scale up again to be able to turn this into a product that you can treat hundreds, thousands of patients. So I think there's going to be a lot about the sort of technical side of trying to sort of improve manufacturing capabilities. But there's so many advances at the moment in stem cell culture in general that I think that there's so many more sort of techniques and strategies that are coming on board now to make that genuinely a feasible objective. As for when, I mean, we already have, so David Gann's group and Blue Rock have already started clinical trials for photoreceptor transplantation. This is a rod and cone transplantation. We are certainly pushing hard to try and get towards clinical trials in the next couple of years as well. So watch this space.
Yeah, is the general goal to create autologous cones from the patient's own stem cells? Or would that even work? Because it's
generally not.
They carry the mutation,
yeah,
right?
It's not to say you can't do it. There are lots of IPS derived or protocols with IPS generated retinal cells. It's more about cost. Because obviously to generate an IPS line for each individual patient, firstly, if it's an inherited disorder, you've got to correct that genetic defect in that patient's cell line first, and then generate your donor cells. If it's not an inherited form, you've still got to then generate and characterize that cell line. And so with that brings a lot of cost. So generally speaking, most teams are working with non-autologous, whether that be an IPS-based or from a cell bank or an embryonic stem cell bank, but either way tends to be non-autologous, really just around cost and being able to test that cell line and know that it'll differentiate exactly as you expect it each time.
Mm-hmm. And if it's non-autologous, is it likely to get rejected? Do the patients have to be on immunosuppressive therapy, like in an organ donation, or is that in the eye not such an issue?
Yeah, I think so. In the eye, less of an issue, as many people know. The eye is often described as a sort of immune privileged space, and particularly the subretinal space where you're transplanting the photoreceptors, in part because photoreceptors are not antigen presenting. So humans, HLAs, they express very little or no HLAs. So the photoreceptors themselves stay below the radar of the immune system. So I think that at the moment, the evidence that we've got so far from a variety of different transplants that have been done, both in monkeys and in patients as well, that it seems to be that it's unlikely to be a major issue. So there have been transplants of human fetal sheet transplants. There's also been cadaveric retina, so from adult donors. That they've suddenly survived. I mean, I think in the studies they were also reporting out to beyond a year. I don't know that they were reporting much beyond that. The same goes for embryonic stem cell derived retinal from organoids. Similarly, has survived for many months, years. And generally speaking, and actually, I think Michiko Mandai's group did quite a nice study looking at ES and IPS derived human derived retinal tissue and showing that it even has potentially immunosuppressive properties and sort of via TGF beta signaling. So I think that overall you're probably going to have immune suppression initially, but the hope is that, for acute, similar to, I think, the RPE transplants that have been done so far, they generally had a short course, but it wasn't sustained. And the hope is that it'll benefit from that immune privilege status.
Okay. Then there's a question about one of your experiments, the one with a light-dark test chamber.
Yes.
So the question was, how long do the animals remain in the chamber? And if you do the tests many times, if there's an habituation that they will spend time more in the light side, even though you would expect them not to if they do see. I mean, you can see that in a wild type, right? Whether they would habituate.
Yes,
how often do the tests and how long do they stay in there?
Yeah, I'm trying to think now. It's a long time ago since we used the test actually as to what the protocol was. I'd have to go back and look at the actual notes. From recollection, it's a few minutes per trial. So I think that there was an initial, you tend to put them, we started recording when they were near the door, simply so that you're not sort of biasing it to start with of one chamber or another. I think they had a brief habituation period, and then we were recording, and as I said, I would have to go back to the paper, I'm afraid, to actually remember the details as to how long, but it's a relatively short period of time. And then we would do another trial another day. So then you still have that exploratory behaviour. I mean, we've done some work that's not a different test, actually, and I'm still trying to write it up for publication, but we were looking at lumen sweep responses. This wasn't actually necessarily in the transplant, but it was more about a comment around habituation. And I think that some of these sort of salient cues, actually, they don't habituate to, and we could actually see that you could test repeatedly and it would still trigger the response. Something like light and dark, yes, I mean, I think, you know, mice can settle down and stop exploring, but we were doing a trial and then we would come back and do another trial another day. So you've still got that sort of impetus to explore.
All right. So next question is a bit longer. I'll just read it to you. What do you think would happen if H cones were transplanted to a mid-degeneration stage of cone loss from a coned-dominant model, like tree shrew or ground squirrel, that have highly structured, dense cone populations, more like the fovea, than seen in the nocturnal rodent model? Would material transfer be beneficial to remnant host cones?
Good question. So you won't, two parts. As far as we know, material transfer is species specific. So we did our first experiments in mouse to mouse. I know Valerie Wallace's group then followed up with looking at human and human mouse co-cultures. And human to human, you can do material transfer. Human to mouse didn't happen. So human cones into a tree shrew probably won't work. Tree shrew cones into a tree shrew, I don't know. I would imagine that material transfer would occur. As I said, we think it seems to be certainly it's happened in mouse, it's happened in human. I would sort of hypothesize it would probably happen in other species, so long as it's self to self. In terms of highly dense, and whether or not you then have migration and material transfer, as I said, when we did it into the intact retina with the mouse, you get some true integration and also material transfer. And when we looked, it was predominantly material transfer when the retina is intact, and then you get a few, around 5 to 10% of cells that actually migrated into the recipient structure. So I'd hypothesize it'd be similar, but I don't know for sure.
Okay. So there are two questions that are asked about the number of cells that you transplant, not transplant, that you're injecting, versus the fraction that actually gets integrated, and how comparable it is, the number of cells, how comparable it is to the mouse, between mouse and the human macular that you would aim for at the end.
Yeah, so we're actually injecting similar, so we inject around 500,000 in the experiments I was showing you. And that actually sort of equates to roughly the sort of density of cones in the macula anyway. So we cover an area in the mouse that covers an area of around 2-ish millimeters squared. So it's about 250,000 per millimeter squared. So it's not far off the macula, which is around 200,000 per millimeter squared. So as a density, it's not a million miles away from that seen in the fovea. In terms of how many survive, it's quite difficult to quantify. We cover a large area, and we get a bit hand waving as to precisely how many. We do actually need to start putting together somewhere we can start to correlate between the number of cells and the function we see. And actually this has come up with a couple of reviewers' comments recently. It's quite difficult because our modes of testing aren't all directly relatable, so we can't follow it every mode of testing throughout the entire process. So histology, optomotor, MEA, etc. We can't necessarily trace them all the way through, and some are limited by what you can visualize. So I don't honestly know the fraction that survive. I would sort of hazard a guess that it's probably more than half of them, but I'm not sure beyond that. I would need to actually do some cell counts.
Okay. Have you looked at the transplants long term, for example, 12 months or so after transplantation?
Yeah, so we've gone out to 9 months, so not far out off, and still see similar cell masses. So we think that they do survive long term. We've actually just set up some to do long term, and to just leave them for as long as we dare, to try and go beyond a year for exactly that reason as well, and try and look even longer.
Mm-hmm. And what is the technique that you actually get such nice layers of cells than rather just a bleb?
Well, I'll say my brilliant surgery, but it's slightly flippant. It is, to be honest, it is around surgical technique and getting a good cell suspension. I think that we've done a lot to work on viability, to have really good cell health at the time of injection. And then trying to obviously, just have a nice consistent bleb formation. And actually, once you inject, the retina then will settle down on top anyway, and then we get spread. I mean, I wouldn't say that it's always identical. Sometimes we get a bit of a strip, and sometimes they're a bit rounder. So there is variation in the shape of distribution if you're looking down on the retina. In cross section, it seems to be fairly well evenly distributed.
Do you know whether your human cones connect to cone bipolar cells only or also to rod bipolar cells? And second, do you think you can influence that with your developmental cues?
So no, we don't know. And actually, that's something that we are investigating at the moment. So we're trying to develop tools to do synaptic tracing to try and see what our donor cells are connecting to, and to be able to try and model this connectome to see how normal it is. And we can also look at electron microscopy, fluorescence EM, and start to sort of rebuild individual cell connections. So those, I think, will be really, really interesting. And I think particularly in light of some of the work that's been done in recent years, looking at disease models and laser ablation and so on. So Rachael, I think I mentioned in the talk a couple of times, Rachael Wong's papers, but there are others as well in rabbit models, primate models, etc., and showing slight differences between how on and off bipolar cells will respond to injury. I think that our histology suggests that both respond, so I'm expecting that both may be involved in the connections, but I don't know that for certain. I think with the only thing that we have to suggest that we might be able to sort of modulate it is with the reintroduction of our guidance cues, that we start to see a better, I mean, this is preliminary data at the moment, so don't hold me to this, but we do see a slight shift in the sort of, as I said to you, we had a predominance of on at the expense of on-off. And with our guidance cue, we seem to get more of the on-off responses than we were seeing previously. So, as I said, it's very much preliminary data at the moment, but it might suggest that there is some difference in sort of reactivating your on and off pathways. So it'd be really interesting to probe it further.
Okay, and another longer question. So is the protocol a 2D or 3D approach? How scalable and GMP compatible is that? And you aim to sort, but you cannot use GFP later on if you're going to a patient.
Yeah.
And if that's the case, how do you isolate or enrich for cones?
Sure. So it's a sort of 2D, 3D hybrid system at the moment. So we use retinal organoids and then we dissociate from there. Is it GMPable? Yes, I didn't actually show the data in this, but we have now converted our processes to fully GMP compatible. So this isn't trivial because obviously a lot of the reagents you use along the way are that there may not necessarily be GMP versions of it. So you have to quite often adapt your sort of culture methods and the reagents that you use. But yes, we have a GMP compatible process. In terms of cell sorting, no, you're absolutely right. We don't use viral vectors to go into the patients. We can sort our cells. That's not in details I can go into as to how we sort them, but we are able to isolate our cones from a mixed population without using viral vectors or GFP.
Okay.
Sorry, we ended up sort of getting into the
yeah, sure. Is there any research on how to limit damages caused by subretinal injection in the foveal region? Or is that not an issue at all?
I probably better ask a surgeon as to how to minimize that. As I said, I'm not a clinician, so I think you clearly have to be careful if you're going to encompass the fovea. I think that the macular, sort of peripheral macular, I think is easier to target, but I'll leave that to the surgeons to answer more fully.
So what kind of immunosuppressive approach was used for successful transportation, if any, at all?
So in the experiments I've shown you with the xenotransplants, so the human cones into the mouse, these are immunocompromised. So it's the nude mouse line, so FOXN1 knockout. So that seems to be very good at supporting long-term survival. So we crossed that with the RD1 and the AIPL1. We have done it into the non-immunocompromised as well. We haven't gone out so long. We've only gone out for a couple of months at the moment and still see survival. We're going to extend this and do a bit more work on that from there. So it's through FOXN1 knockout.
Okay. Then question: synaptic connections in the macula are extremely precise with a 1-to-1-to-1 connectivity. From cones to bipolar cells and ganglion cells, and do you think it's realistic that this will be recreated with this approach? And if not, if you have not a one-to-one connectivity, do you think if it's still useful to obtain a real functional benefit?
I think that yes, it is still going to be beneficial even if you don't achieve that ratio. How close we'll get, I don't know, and I don't think any of us know until we go into models that have maculas developed to test that, whether or not that's in primate or going into clinical trials. But I think it is realistic to achieve a reasonable connectivity, and how close to that, I couldn't say at the moment.
Yes. We already went over time, so I have one last question. Do you know what kind of cones are integrating in the retina? For example, you have S-opsin-rich cones or the M or the L cones. Do you know which ones actually integrate better?
So we haven't compared. We primarily sought L and M. So because obviously those are the predominant ones in the macula as well. So we have focused on that. So we haven't actually compared between the two directly. So, I mean, it would be interesting to do a mix and to see if there is a difference in terms of which ones connect. But our system is biased towards the L and M. Okay.
Then I think we're through. We're probably getting more questions, but time really is up. Rachael, thank you so much. It was very impressive, all this body of work that you've shown us. And I thank the audience. I thank for the wonderful participations and the good questions. And hope to see you next time for our next Journal Club. Thank you again, Rachael.
Thank you.